So it's phenol chloroform. The first step is to prepare phenol. So preparation, phenol solution. This can be done in two different pH. So if we want to extract DNA, we have to prepare phenol at pH 8. And to do that, we will take about 5 grams, for example, of phenol, and we will mix it with 5 ml of tris. Normally, tris can be used in concentration of 50 millimolar pH 8. After mixing these two solutions, it is important to incubate the mixture in room temperature for about 1 hour. So this will lead to form two layers. If you mix the solution well, so after dissolving the phenol with the tris, if you leave the phenol for about 1 hour, you will get a tube like that. Sorry. There will be a phase separation. So all phenol will be at the bottom layer, while the tris will be at the top layer. So after 1 hour, you will remove all the tris. So this will discard. And then you will add again 5 ml. So that will be the tube. That will be the phenol in red, which is the normal color. And then you will add again 5 ml of tris. Mix it well. Leave it again for 5 minutes. And after the 5 minutes, again, there will be a phase separation. So that's the phenol, and that's the tris. And again, wash the phenol with the tris by discarding or removing all the layer of a tris, and then adding the same volume of a fresh tris pH 8. So this repeats about 5 times. After 5 times, you can measure the pH of the tris after the phase separation in the 5 times. Here. And you can see that the pH of this buffer will be 8. So now the phenol is ready to be used. Normally what I'm doing in my lab is mixing 1x of phenol with 1x of chloroform. And after that, I will take 1x of cyanlysate, which containing the nucleic acids. So that's the cyanlysate containing nucleic acid plus proteins plus other components of the cells, such as lipids, such as carbohydrates, and other mineral salts. Okay, so for example, if we take 5 microliters of cell lysate, this will be mixed with 5 microliters of phenol plus 50 microliters of chloroform. And then mix it well, mixing by pipettes. Centrifuge the sample, spinning, and you will get 3 different layers. So if we have this tube here, the microfuge tubes, you can see clearly that after centrifuge for about 5-10 minutes, it's enough, you will find here the red layers of phenol, you will see a ring of white color, and this is normally the coagulated proteins. And at this top layer, your sample contains a mixture of DNA plus RNA. And in order to remove the RNA, you have to use RNase. So, after this step, we will take only the top layer, and again we will mix it with 1 volume of phenol and chloroform. And by repeating that about 3 times, you can get a very clear sample of, so basically you will take the phenol chloroform, you will mix it again with the top layer, which is containing the nucleic acid, and after mixing that, you will centrifuge it, you will get also the phase separation, but at the second time you will lose the coagulate, the ring layers, or the ring layers will be very weak. And remember that every time by repeating the phase separation using phenol chloroform, you will lose some amount of nucleic acid, but you will get a high purity of the sample. Remember, the phenol is quite dangerous. It can cause skin burning if the phenol being in contact with your skin, and it also has a very discomfortable smelling that can cause harmful effects in your respiratory system. So you have to wear gloves, you have to wear also goggles and mask. So that's the end, and please, if you enjoy my YouTubes, don't forget to subscribe the channels. Thank you very much.